Hi, my name's Anna. Today I'm going to be talking about horizontal gene transfer in marine environments. Genetic change is the driving force of evolution, and evolution is the driving force of adaptation, allowing us to keep up with an ever-changing environment. So genetic variation can occur through mechanisms like mutations, recombination, chromosomal rearrangement, and horizontal gene transfer. So mutations are when within a base pair something might be incorrect, whether in one location or more is inserted or deleted. Recombination is common in meiosis, where chromosomes cross over and their legs kind of break off, meaning that every time they reproduce, there's slight genetic variation within the daughter cells compared to the parent cells. There's also chromosomal rearrangement, where parts of the chromosome might be deleted, duplicated, inverted, or translocated. And of course, there is horizontal gene transfer. Cells can inherit DNA vertically from parent to offspring during reproduction or horizontally through the transfer of genetic material between organisms. And although it's less frequent than vertical gene transfer, horizontal gene transfer can drive rapid adaptation by enabling organisms to acquire new or novel genetic traits. So horizontal gene transfer occurs through mechanisms such as transformation, conjugation, and transduction. Transformation is when bacteria takes the floating DNA from their environment. Sometimes it can come from a dead cell or from a lice cell where the bacteria is just kind of floating around. There's conjugation that involves a direct transfer of genetic material through cell-to-cell contact, typically from a host to a recipient, and it's done through a bridge called a conjugative pilus. And you can see it here next to C. And then there's also transduction. Transduction is a virus-mediated way to transfer genetic material. It uses bacteriophages to transfer DNA between the cells, which you can see in B. So horizontal gene transfer occurs everywhere, including the ocean. Beyond the classical mechanisms I just went through, recent research has revealed that there are additional pathways and vehicles that can facilitate gene transfer, especially in complex natural environments. So 3 methods that are hypothesized to mainly facilitate the horizontal gene transfer within a marine environment is marine viruses, gene transfer agents, and extracellular vesicles. And you can see in the top right corner basically kind of a visualization of all 3 of these things, but I'm also going to go through them all one by one. So marine viruses transfer their own DNA or mispackaged cellular DNA to an organism during infection. However, they are typically limited in the type of host that they can infect. They need to be kind of similar to the original cell. There's also gene transfer agents, which are virus-like particles that package and transport random fragments or host cellular DNA. And if you look at the bottom, GTAs are very similar structurally to bacteriophages, so it's similar that they have a similar function and way of going about. There's also extracellular vesicles. These are small, typically 50 to 200 nanometers, membrane-bound sacs released from the surface of a cell. So if you look in the bottom right corner, you can see there's actually a vesicle budding off of one of the bacterias. It basically just kind of pinches off, and inside of it is different materials such as DNA, RNA, proteins, and other biological molecules that can then be transported to other cells. So there is currently an increased rate of horizontal gene transfer in the ocean, things that it has increased than previous things, and they believe it's because of increasing water temperatures and increased ocean acidification. Warmer water temperatures are accelerating the microbial metabolisms and reproduction rates. And so gene transfer depends on ecological connections between organisms. As we saw previously, sometimes it literally means a connection side by side in order to transport this genetic material. And with more microbial activity in response to this increasing temperature and warmer waters, there's an increase in the frequency of contact and genetic exchange that is between marine bacteria. Also, there's acidification. Acidification is contributing to a lower pH, which means that more bacteria are in a stressed state. And when bacteria are stressed, they are induced to release genetic material into the environment, and it also makes them more susceptible to taking up foreign DNA. This is a survival mechanism that they have in order to rapidly adapt to changing environments, and there have been multiple studies that confirm that this is how bacteria is reacting. But this is increasing a rate of horizontal gene transfer. So now that we know what's happening, what are the functional genes that are typically being transferred? The first is, they're typically what you would expect. They would be genes that help with metabolism, help with nutrient acquisition, they help with stress response, and they also happen to help with antibiotic resistance. These are all useful for an organism to stay alive and to reproduce, but they don't always have positive consequences for all the other life forms that rely on the ocean, for example, us humans. So a subtype of the genes that are being transferred are called antibiotic resistant genes, or ARGs. And so they basically are taking harmless bacteria and turning them into a pathogen whose infection is really hard to prevent and treat. And so this is partially due to an increased use of antibiotics in aquaculture, in addition to, again, global warming and increased pollution. And so this is creating an enhancement of drug-resistant bacteria. So for example, Vibrio harvei, you can see it here in the top right corner. It's actually one of the bioluminescent bacteria that I believe we've talked about in class. And so there has been more of this drug-resistant and multi-drug-resistant bacteria found in fisheries all around the world, especially in seafood farms and in shrimp farms, and they've been found in South and Southeast Asia, in Europe, and in North America. So as I was saying, it increases its pathogenicity. It's taking something that was once harmless and is now giving it a bunch of things that makes it not so harmless. And so things that contribute to it is virulence factors. Virulence factors include things like proteases, hemolysins, phospholipases, and exotoxins, and then different secretion systems. The emergence of ARGs is one of the reasons that horizontal gene transfer has recently become a significant topic of discussion amongst biologists across different specialties. So more research has been dedicated to understanding how to prevent the spread of these types of genes, but also to understand what is promoting the spread of these genes. And so one of the things that they have found was that an increase in plastic pollution is contributing to it. So microplastics have actually been identified as a hotspot for a concentration of pathogens because they provide a surface to facilitate high-density microbial interactions. Basically, a lot of them can kind of stick to this microplastic. It introduces the horizontal spread of these antibiotic-resistant genes, and so therefore making it a hotspot. So a way that you can visualize this is if someone's sick, goes to a party, and they're surrounded by a lot of people, that party becomes a hotspot where the germs are then spread and everyone gets it. And so, again, these microplastics are increasing the contact rates between different diverse microbial species, and they're all picking up this antibiotic resistance gene. Also, increased acidification and the rise of hypoxic zones in marine environments have created areas that favor the proliferation of pathogens. So facultative anaerobic bacteria like Vibrio and Archobacter are both marine pathogens, and so when all the other life forms are starting to die out or move away because of the acidification and low oxygen zones, these bacteria are starting to thrive, which then is not in turn making it good for all of us. A research group from Tianjin University of Science and Technology was wanting to look and prove that hypoxic conditions actually increase the number of pathogenic bacteria, and so they did an experiment to prove this in the Central Bohai Sea. What they did was, so in the Central Bohai Sea, it regularly develops into a seasonal hypoxic zone, typically in the summer. And so they wanted to follow the oxygen concentration and also the different bacteria that is in the zone and seeing if the bacteria changes over time. And so using a continuous time series sampling across multiple depths, they were able to confirm that the dissolved oxygen levels decrease in the summer and reach their defined hypoxic threshold, which was less than 2 milligrams per liter. And so during this monitoring period, they also collect and sequence the microbial communities that were present. They also looked at the phosphate concentrations. And what they wanted to see is if these bacterial communities change as the oxygen drop. And what they found was that the entire microbial ecosystem does in fact shift. This is the results of what they were able to sequence. So again, reminder, Shannon diversity is species richness and KO1 is total richness. And what this figure shows is that the Shannon index continually increased from May to August, while the KO1 index followed an inverted U curve. The KO1 index showed that richness was significantly higher in July than it was in May and June, with a decrease in August. And so for the samples found in the bottom of the water, the Shannon index increased from May to July, but decreased in August. And the KO1 index also had a lower values in August. There were also some statistical analysis done. The Mann-Whitney test showed that Shannon, Simpson, and Pilu were significantly higher in the high oxygen sample than in the low oxygen sample, indicating that some of the species could not tolerate the decrease of oxygen concentration in the hypoxic zone. And so what was left behind was the pathogenic bacteria. So in conclusion, overall, horizontal gene transfer is a very important concept to understand and monitor in order to predict and promote a healthy marine environment in the future, and it's something that we should always keep an eye on. Thank you.